Hola, hola. Sí, está bien. Ok. Ok, voy a decir tres cosas en español antes de empezar. Muchas gracias por venir. Como tenemos un invitado internacional, que es la profesora Lappas de la Universidad de Melbourne, todo este día va a ser en inglés, pero la idea es que, como está dentro del contexto de un curso de posgrado, que los estudiantes participen y todo, y que el lenguaje tampoco sea un impedimento. Entonces, si tienen preguntas en español, no se sientan intimidados en preguntar, y entre todos vamos a poder traducir y hacer esto un poco más dinámico. Eso. Okay, so hello everyone. Good morning. Thank you for coming to this symposium. Before to start, I would like to just mention some important things. For example, our safety precautions. The exit. You have one exit here and another exit in your back. So please be aware where are the exits in case that something happens. Hopefully no earthquake, by the way. Just be aware that is the exit, okay? Also, I would like to acknowledge the contribution of the different faculties that allow us to perform this symposium. The Faculty of Pharmacy and also the Faculty of Biological Science here at the University of Concepcion. Also the Fondesit, because thanks to the funding agency, we can also invite our speakers today as well. Okay, so we're good? Yeah, okay. Acknowledge, exit, all good. So, well, thank you, everyone. As I've mentioned in the flyer, so this symposium is about extracellular vesicle. I lost the pointer already. It's over there. Yeah. Okay, so the symposium is about extracellular vesicle, and the idea is to discuss a couple points on the extracellular vesicle field. The idea is to discuss, of course, the different type of extracellular vesicle during this day, mainly focusing exosome, and hopefully when you finish the symposium, at least you will know that exists different type of extracellular vesicle, and not all the extracellular vesicle called exosomes. And for that, we have 5 speakers. We have Dr. Marta Lappas from the University of Melbourne. Then we have Dr. Luis Sobreria from the Pontificia Universidad Católica de Chile. And both will talk about their research line mainly in pregnancy, but also some data about exosome on also extracellular vesicle. Then we have, according to our program, we have our coffee break, and then after the coffee break, we have Dr. Andrew Quest from the University of Chile to talk about also extracellular vesicle and cancer. And then we have our lunch break, and afternoon we have two more speakers, Manuel Varas from the Universidad de los Andes, who will talk about exosome extracellular vesicle in the context of ovarian cancer. And finally, Cristian Carvajal from the Universidad Católica de Chile, who will talk about extracellular vesicle in hypertension as well. So you can see it's different topics. We have all a common interest in exosome and extracellular vesicle. So, of course, I would like to just give you a 5-minute introduction about the context of extracellular vesicle and the exosome. And this is a hot topic in the literature. And it's plenty of information about the potential role of this extracellular vesicle, mainly cell-to-cell communication. So I just put this slide to highlight the explosion in terms of the research on extracellular vesicle, okay? But even if we already know that there are different types of extracellular vesicle, we know also that this extracellular vesicle, we have different subpopulation of vesicle and probably different function. It's a lot of gap in terms of the basic knowledge about the biogenesis, the heterogeneity of this extracellular vesicle, the content of this extracellular vesicle. So even if there's a lot of research going on in terms of the extracellular vesicle of exosome, it's a lot of very important basic questions that need to be answered as well. So to just put everything in a context, I have this simple and a little bit fancy video about extracellular vesicle and why this extracellular vesicle is like a hot topic now in the literature. So you can identify extracellular vesicle in general in all the biological fluid and also in plasma, in circulation. And they are a hot topic now because they function as a transporter, like a carrier of information. They are like letters in which you can put information on it, and one cell will release this vesicle, which contains specific information that seems to be a metabolic representation of the originating cells. And then this vesicle will interact with other cells, release the content, and modify the biological function of the target cells. So that is the basic concept about extracellular vesicle. So you have one cell that have the capacity to communicate with other cells, send a specific message inside of this vesicle. This message can be a specific proteins, can be a specific microRNA, because they are rich in microRNA, and modify the phenotype of target cells. So it's a lot of information, mainly in cancer, about how this exosome can prepare the pre-metastatic niche and prepare the invasion of cancer cells. So in terms of you just do a quick searching in PubMed, you can see this is the number of publications per year, and you can see how is the explosion of this publication in the last 10 years. Okay, so it's a hot topic in the literature. And so what we know in terms of the biogenesis of exosome. So this is just a cartoon with the cells, and you can see that the exosome, they are coming from endocytic compartments. And this is the main difference with other type of extracellular vesicle that is coming mainly from the plasma membrane, like microvesicle. So the exosome, they have a different origin. Also, people can separate the vesicle according to the size. There's a lot of information about the size of microvesicle, and they are bigger compared to the exosome. Exosome, the size is more uniform, around 100 nanometers. But I think the size is not that important, and probably the different origin of this vesicle is the main difference between exosome and other type of vesicle. So we know that the exosome can circulate in the cytoplasm as intraluminal vesicle. In some point, this intraluminal vesicle will be incorporated in multivesicular bodies, this structure, and then traffic into the plasma membrane, bind with the plasma membrane, and release this exosome. And we also know it's a lot of information in cancer and some information also in pregnancy, in which seems to be that these cells can sense the microenvironment, for example, and the low oxygen tension, which is a common characteristic of tumor microenvironment, the release of these vesicles are higher, and also the content is different. And also we have some information in diabetes condition in which the exosome, the number of exosomes are higher, and also the content is also different. Then this exosome will be released to the microenvironment and then interact with the target cells. Is different type or different option of interaction, and then release the content and modify the biological function of the target cell. One of the bigger questions in the field is about the targeting also of this vesicle. So this is a cartoon, but in terms of the physiology, when one cell releases exosome, this exosome can interact with many other target cells. So one of the biggest questions in the field is if this vesicle, they already have a predetermined target in which they know what are the organs of tissue that they're going to interact with. And we don't know the answer, but it's a beautiful paper published in one of the big journals in the field, I think it was Nature, in which the author shows that if you change the profile of integrin in the membrane of exosome, this profile of integrin can also regulate the targeting of the exosome. But we don't have much more information about that. If you don't like the cartoon and you want to see a real image, this is a beautiful electron microscopy image about the exosome. You can see the exosome here. This is the plasma membrane. You can see here the multivesicular bodies and the exosome inside. Okay? So you can see they are a small vesicle, and then they are released from the plasma membrane. So in terms of the size, you can see that the exosome usually is around 100 nanometer size, and the microvesicle, they are bigger. And it's important also to point out that it's a lot of information, especially 5 or 10 years ago, about the morphology of the extracellular vesicle, and they believe 5-10 years ago they were like a cup-shaped morphology, but we know now that the cup shape is more like an artifact based on the sample preparation for the electron microscopy. So you can do a cryo electron microscopy. You can see they are a circle, the exosome and the other extracellular vesicle as well. So how you isolate the exosome is just a little bit of background. You can isolate exosome from cells that include primary cells or cell lines from the cell conditioned media. You can isolate the exosome, define the content, and also take this exosome, interact with other cells. A couple of big common questions when you isolate exosome from cells is about how many cells you need to get something, enough amount of exosome to perform your experiment. I think that is one of the bigger questions in the field. Of course, the isolation method also is important. Also, you can isolate exosome and other type of vesicle from biofluid, plasma serum. So they are very attractive as biomarker because they are small, so they are very stable in circulation, and they can transport specific information, microRNA and protein. So as the content is a representation of the originating cell, they are very attractive as biomarker discovery. And also you can identify in saliva and other biofluid. Using a similar approach, you can isolate the exosome and then characterize the content and do some bioactivity assays. So what is the key when we work with the extracellular vesicle? Probably is the isolation. Just I would like to point out that the exosome is a specific type of vesicle in which when you are working with the vesicle, also you need to perform at least 3 or 4 different experiments to demonstrate that you are working with the correct number, with the correct type of vesicle. It's just, this is a table, it's published and available online, and you can see the different methods to isolate exosome. And every single method is different, and probably every single method also can isolate different type of vesicle. It's something that you also need to keep in mind. This is a cartoon in which you can see that it's not a perfect method to isolate exosome, and every single method have pros and cons. And probably you can, of course, there are some methods that are very time consuming in which you can have a better purity of the sample, but the yield is lower. So it depends what is your aim. If you're looking for proteomics or profile the content of this exosome, probably the purity of the sample is important. If you're looking for exosome for injecting animals or perform some in vitro experiments, I think it's difficult to get good purity of the sample because probably you're not going to get enough to perform your experiment. So you can see it's a lot of variability in terms of the isolation of the exosome. So the only advice that I have, and hopefully we can discuss this during the day, is whatever method that you're using, trying to be consistent, use the same method all the time. This is just a paper that we published earlier this year about we described 3 different methods to isolate exosome that you can take a look if you are interested. Also, the exosomes are very attractive for diagnostic. So you just have a table in which is available online as well, in which you can see that exosome can be isolated from different biological fluid, and then can be used as biomarker from different pathological conditions as well. Okay. In terms of the use as exosome as therapeutics, biomarker discovery, and also as a therapeutic approach, if you're going to the clinical trial website from the NIH, you can see it's around 97 different clinical trials now going on in terms of the exosome. So when people think that it's not much about exosome, well, I think we know enough to perform clinical trials, actually. So it's 97 clinical trials going on in different pathological conditions involve the exosome in the experimental, in the trial design, okay? Also, the exosomes are very attractive for therapy, but of course, you need to check your doses, check what is your model, your animal, also the content to check what is the fed, and the mechanism associated with your fed are important as well. So finally, just keep in mind for the rest of the day that exosomes, they are a specific type of vesicle. They are different compared to the microvesicle. So we have some tools to characterize the exosome and other microvesicles as well to make sure that you're working with the correct type of vesicle. And the exosome, you have the capacity to isolate exosome, for example, coming from tumor cells. You can use as an early detection from different pathological conditions. The cartoon is in the context of ovarian cancer, but also you can use it a drug delivery system in which you can incorporate drugs and other molecules in the exosome and use as potential therapeutics. Also, as you can get exosome from biological fluid, they are like a liquid biopsy. So you can also track the real-time monitoring system of different pathological conditions using the exosome as well. Okay, so that was a very brief introduction about the exosome, and now I will introduce our first speaker. Okay, first of all, I would like to just thank Marta, who came all the way from Australia, from Melbourne to Concepcion. So thank you for taking the time to be with us. And she has been here for essentially the whole week, so I think it's a great opportunity for a student as well. So Marta Lappas is Associate Professor of the University of Melbourne in Melbourne, Australia. And she's the head of the Obstetrics and Nutrition and Endocrinology group at the Mercy Hospital in Melbourne. She is a worldwide expert in inflammation during pregnancy, mainly in preterm birth and gestational diabetes. She has published already around 180 papers, and mostly in good journals, and obtained funding for more than $4 million in the last 5 years. So thank you, Marta, for being with us, and we're looking forward for your presentation. That's for you. No, you need to just hold this.
Hold it.
Yeah. You can put it.
Thank you. Thank you for the invitation to present today. And it's fantastic being in Chile for the last week. I almost feel like I'm a Chilean. So today what I want to do is talk about some of the work we've been doing over the last few years on inflammation in pregnancy, and more recently some of the work I've been doing with Carlos on the potential role of exosomes in pregnancy. But before I do that, what I want to do is introduce the university that I work for. So that's the University of Melbourne. So it's a very big precinct in the city centre. It's the oldest and most prestigious university in Australia. It's got, in addition to the university, it's associated with 8 different hospitals, including cancer hospitals and a few maternity hospitals as well. It's also affiliated with many leading research institutes, including the Peter Doherty Institute, which mainly focuses on immunology. Now, it is, as I said, it's the most prestigious university in Australia, and it's ranked number 32 in the world. So the department that I work for is the Faculty of Medicine, Dentistry and Health Sciences. It's got almost 2,000 staff, and most of these staff are research staff. And it also has lots and lots of students, including greater than 1,300 students who are doing masters or PhDs. There's many different themes associated with our faculty, including ageing, cancer, neuroscience and psychiatry, and a field that I work in, which is women's health. And in terms of women's health, we look at complications of pregnancy, as well as various reproductive health, such as endometriosis and menopause, and it's quite multidisciplinary in scope. There are basic scientists, there are clinicians, and epidemiologists as well. So I work at the Mercy Hospital for Women, which is about 20 kilometres from the main campus. This hospital is quite new, and it's co-located with probably about another 10 different hospitals and research institutes. And this is just our laboratory here. And my group, as Carlos said, is the Obstetrics, Nutrition and Endocrinology group. And I have many, many focuses for my research, particularly preterm birth and gestational diabetes and obesity. And these are just some of the samples that we collect, including placenta, myometrium, decidua, maternal and cord bloods. We also collect adipose tissue and skeletal muscle. We also do some studies in depression in pregnancy, where we're looking at the impact of depression and antidepressants on fetal outcomes, as well as a long-term gestational diabetes follow-up project where we are trying to predict the development of type 2 diabetes in women who've had gestational diabetes. And these are the fantastic midwives that we have who collect all our samples for us. And so our system that we use is it's called REDCap, and when the research midwife consents the patient, she obtains all the clinical information on the mum and the baby in real time, and they are put into the system, so we have all that information immediately. So that's just a bit about our group. And in terms of my talk, so what I want to do is talk about the pathophysiology of gestational diabetes. I'll then introduce the role of inflammation in gestational diabetes. In particular, I'm focused on the role of adipose tissue in disease pathophysiology. So I'll talk about adipokines and some of the recent work we've been doing on exosomes from adipose tissue, and then how we can prevent gestational diabetes by targeting inflammation. So gestational diabetes is defined as any degree of glucose intolerance with the first onset or first recognition during pregnancy. And what is quite alarming is that GDM is on the rise, and this is in concert with the obesity epidemic. In our hospital, it complicates about 20% of all pregnancies. So it not only impacts the mum, but what is also distressing is that it can have long-term effects for the offspring. So these offspring are usually born bigger, they have increased fat mass and adiposity, and this predisposes them to a number of diseases later in life, including diabetes, cardiovascular disease, and obesity, and also certain cancers. So there's a huge, huge economic cost associated with gestational diabetes. In pregnancy, caring for a woman with gestational diabetes costs about 35% more than it does for other women. Post-pregnancy, 2 to 5 years post-pregnancy, it's also showed increased costs of about 20%. In addition, there's also the long-term costs of gestational diabetes in terms of both mum and the offspring, and these costs are incalculable. So what is really, really depressing is that the current treatment and preventative strategies for gestational diabetes are really quite unsatisfactory. And insulin, which is commonly prescribed for gestational diabetes, is really fantastic in regulating mum's blood glucose levels. However, it doesn't protect the fetal offspring, adverse effects on the fetal offspring. In addition, there's also some recent evidence to suggest that insulin therapy may also be associated with increased risks of developing type 2 diabetes later in life for the mum. There is certainly some very promising data on diet and exercise in preventing gestational diabetes. However, this is really only effective in high-risk groups. It's also complicated because people have their own certain diet preferences, as well as there's compliance issues. So clearly we need a better therapy to prevent gestational diabetes. However, to do this, we need to better understand the pathophysiology of gestational diabetes. So gestational diabetes is characterised by insulin resistance. Now, insulin resistance is a normal part of pregnancy, and this happens usually in the 3rd trimester, and it's important because what it does, it directs the nutrients away from the mum towards the placenta. The placenta can then transfer these nutrients to the fetus to allow for adequate growth of the fetus. In women with gestational diabetes, however, this insulin resistance is much more severe, which results in increased placental uptake of the nutrients and therefore accelerated fetal growth and development. So our studies over 10 years ago showed that in adipose tissue and skeletal muscle of women with gestational diabetes, the insulin-stimulated glucose transport is suppressed. There's reduced expression of the insulin signaling pathway components, as well as reduced expression of glucose transporters. In concert with maternal peripheral insulin resistance, GDM is also characterised by changes in placental nutrient metabolism. So again, in normal pregnancy, amino acid activity, nutrient transport activity is upregulated, and this again is to allow for optimal fetal growth and development. However, in women with gestational diabetes, this nutrient transport, particularly amino acid system A activity, is increased, leading to higher uptake of nutrients. There's also evidence that there's higher accumulation of lipid droplets in the placenta, and also increased expression of fatty acid transporters, again leading to increased nutrients to the baby. So what we think is that inflammation regulates insulin resistance and and placental nutrients transport, and what I'll do is present some of our evidence. So what we know is that pro-inflammatory cytokines, they increase during gestation. They are predictors of high insulin resistance during pregnancy, particularly adipokines such as TNF-alpha. And they also correlate with fetal adiposity, again, particularly adipokines such as leptin. We know that individuals with gestational diabetes have increased leukocyte counts and also an altered adipokine and inflammatory biomarker profile. Our studies have shown that placenta and adipose tissue and skeletal muscle contribute to this increased inflammatory state by synthesizing and secreting a number of pro-inflammatory mediators, and these are dysregulated in gestational diabetes. So these adipokines, such as interleukin-1 beta, interleukin-6 and TNF-alpha, have been shown to induce maternal and placental inflammation. They can cause maternal skeletal muscle and or adipose tissue insulin resistance, as well as altering placental nutrient uptake, and I'll just show some of that evidence now. So this is some of our data published a few years ago, demonstrating that what we have here is the concentration of the pro-inflammatory cytokine interleukin-1 beta being secreted from adipose tissue. So we have normal glucose-tolerant women here, women with gestational diabetes, either diet-controlled, controlled with insulin, or combined. And as you can see, the secretion of interleukin-1 beta is increased in adipose tissue from women with gestational diabetes. This secretion is dependent on the inflammasome, as we have increased expression of caspase 1 in these same samples, and caspase 1 is involved in the activation of interleukin-1 beta. So then given that interleukin-1 beta 1 was increased in GDM, the next question we asked is, can it cause defects in the insulin signaling pathway in skeletal muscle? So we incubated skeletal muscle with interleukin-1 beta and looked at the expression of insulin signaling pathway components, and found that indeed it could suppress the expression of phosphorylated insulin receptor substrate 1, And this was associated with decreased expression of the glucose transporter, GLUT4, and this resulted in decreased glucose uptake in skeletal muscle. So studies have also shown that cytokines or adipokines can also alter nutrient transport, and these studies are mainly done by Professor Thomas Jansen and his group. And so they've incubated placental cells with, in this case, either TNF-alpha in white or interleukin-6 in black bars, and shown that these cytokines can increase system A amino acid uptake in placental cells. They can also increase fatty acid accumulation in placental cells, and so this is just some data showing that interleukin-6 can increase the expression of fatty acids. So what causes this inflammation? Well, we know obesity is one of the main causes. However, this only accounts for 30% of cases of gestational diabetes. So what are the other causes of the inflammation? Now there's some evidence that infection can be involved in the pathophysiology of gestational diabetes. So bacteria and viruses can activate toll-like receptor signaling to induce insulin resistance and changes in placental nutrient transport. There is some evidence that women with bacteria or virus infection during pregnancy have increased risk of gestational diabetes. In addition, maternal peripheral leukocytes have increased levels of toll-like receptor 4 expression. So the question then we asked is, then can viral products or bacterial products induce inflammation, cause skeletal muscle insulin resistance, and also alter placental nutrient transport? So what we did here was to look at the effect of a viral product, poly-IC, and that activates TLR3 signaling pathway, and found that incubation of adipose tissue and placenta with poly-IC could increase the expression and secretion of pro-inflammatory cytokines from these tissues, suggesting increased inflammation. We then looked at the effect of poly-IC on the skeletal muscle insulin signaling pathway, and again found that incubation with this TLR3 ligand could suppress the insulin signaling pathway components, GLUT4 expression, and then glucose uptake. We then looked at the effect of viral and bacterial products on amino acid transport in trophoblast cells, and again found that these products could, so we've got here TLR3 signaling, and this is LPS, which is TLR4 signaling, found that incubation of our placental cells with viral or bacterial products could increase the expression of amino acid transporters and amino acid uptake. So that's our work on inflammation. But in addition to secreting adipokines, adipose tissue also secretes exosomes. Now, Carlos has spoken quite nicely about exosomes, but what I want to stress is that exosomes are really quite exciting because they are representative of the cell that they came from, and then those exosomes can then travel elsewhere, release their cargo, and cause defects in the other tissues. With respect to exosomes and gestational diabetes, Carlos and his team have done the majority of the work in this. And what they found is that placental exosomes under hypoglycemia or gestational diabetes, the release is increased. There's also increased levels of exosomes in maternal plasma of women with gestational diabetes. And interestingly, the pathways that they target are energy production, inflammation, and metabolism. However, what was quite interesting was they were not attributed solely to the placenta, which is thought to be the main source of circulating inflammation. So they weren't attributed to the placenta because the ratio of placental exosomes in GDM was actually lower. This suggests that there are other tissues that may be contributing to these increased exosome content in women with gestational diabetes. So it's possible that adipose tissue derived exosomes may also be contributing to these exosomes. So therefore, our overriding hypothesis is that adipose tissue exosomes from women with gestational diabetes carry a specific set of proteins, and these may influence placental function. So to test this hypothesis, what we did, we took adipose tissue from 140 pregnant women at the time of term cesarean section. We had 70 with gestational diabetes and 70 normal glucose tolerant women. We did adipose tissue explants, we collected the media, and then exosomes were isolated according to methods optimised in Carlos's lab. So firstly, what we did was to characterise the exosomes, and as you can see, the majority of the exosomes are in the 100 nanomolar range. Exosomes were also characterised according to the presence and absence of exosome-specific markers. So what we have here is 3 specific markers of exosomes, CD63, CD9, TSG101, and they were present in the exosomes. And here we have GRP94, which is an endoplasmic reticulum protein, and this was absent in the exosomes, showing the purity of our extraction. And again, we have the shape of the exosomes. So what was quite interesting is when we compared the profile of normal glucose tolerant women compared to women with GDM, we found an increased content of exosomes in adipose tissue, in exosomes derived from adipose tissue. Now this is quite interesting actually because, as I said before, women with gestational diabetes have increased circulating levels of exosomes which are not due to the placenta. So this data suggests that the increased content of exosomes in GDM women may be coming from adipose tissue. We then looked at the relationship between adipose tissue-derived exosomes and clinical markers of, or clinical characteristics of the patients. And what we found, which was very, very exciting, was a significant and positive relationship with exosome content and fetal birth weight. Now this is really exciting because, as I said, fetal gestational diabetes is associated with offspring with increased fetal birth weight. So this suggests that exosomes from adipose tissue may be a link between placenta and nutrient growth. We then looked at the protein profile of the exosomes from adipose tissue, and here's a volcano plot, and of the there was 127 proteins that were significantly different between the two groups. There were 110 that were upregulated in the GDM exosomes and 17 that were downregulated. And what was quite interesting is that the pathways that were associated with these were involved in cell and nutrient metabolism, and they included those involved in mitochondrial dysfunction, mTOR signaling and sirtuin signaling pathways, and we know that these pathways are involved in not only inflammation but also in nutrient metabolism. So then the next question we asked is, do adipose tissue-derived exosomes also regulate genes associated with glucose metabolism in placental cells? So to address this, we took adipose tissue-derived exosomes from normal glucose-tolerant women and women with gestational diabetes, and we added these to placental cells. We did a glucose metabolism PCR array and found that there was probably about 45 genes that were differentially regulated in the two groups. And quite interestingly, the pathways that were the most significantly regulated were those involved in glycolysis and gluconeogenesis, again demonstrating the link between adipose tissue-derived exosomes and fetal growth. So this is our working model. In gestational diabetes, you have sterile inflammation, bacterial and viral infection. This activates the adipose tissue to secrete adipokines and exosomes. They can cause maternal peripheral insulin resistance, leading to increased levels of nutrients in maternal blood. They can also increase the expression of nutrient transporters in the placenta. The placenta can then take up these nutrients, leading to increased babies with increased fetal weight, and then leading to the long-term effects of gestational diabetes. So we think that inflammation, particularly from adipose tissue, is quite central to this process. So the next question we asked is, can we target inflammation to prevent gestational diabetes? So I just want to briefly talk about some of the work that we've been doing on polyphenols. So polyphenols are bioactive components found in fruits and vegetables and nuts and seeds. And we've known for a long time that a diet rich in these foods is very protective. There's also some evidence to suggest that a high intake of the plant diets in pregnancy is also associated with improved pregnancy outcomes, including gestational diabetes, and there's some evidence using Mediterranean diet and a DASH diets, and these are diets high in these fruits and vegetables. Some small trials in gestational diabetes have shown that they can improve levels of insulin, inflammation and oxidative stress. However, we don't know what contribution polyphenols have to these positive effects. So that was an aim of our project. So we are doing, there's two ways we are assessing this, and that's using in vitro and in vivo model of GDM. So in vitro, we take placenta, adipose tissue and skeletal muscle from pregnant women. We use TNF-alpha to recapitulate inflammation and the defects in the insulin signaling pathway associated with GDM. And in our in vivo mouse models, we use two mouse models of gestational diabetes. One is the heterogeneous leptin receptor gene knockout model, and that's a model of insulin resistance. And we use an adiponectin gene knockout mouse model, and that's a model of beta cell dysfunction. Now, these models are really quite neat, because when these mice become pregnant, they spontaneously develop gestational diabetes. In addition, the offspring have also got increased weight, which are the maternal and fetal characteristics of gestational diabetes. So then what we've done is to use various polyphenols to see their effects on the pathways involved in gestational diabetes. So here we've got some data in skeletal muscle incubated with two different polyphenols, nobiletin and naringenin, and they are primarily found in citrus fruits. And so what we can see here is we've got a Western blot showing that TNF-alpha, as expected, decreases the expression of the GLUT4 in skeletal muscle, and this effect can be alleviated by pre-incubation with polyphenol nobiletin. Here we've got glucose uptake in our skeletal muscle, again demonstrating that pre-treatment with our polyphenols can alleviate the decrease in glucose uptake mediated by TNF-alpha. In vivo, so these are in our mouse model, this is the leptin receptor deficient model. We have from gestational day 10 to gestational day 18, the mice receive oral gavage of our polyphenols, and then we do on gestational day 17, we do an oral glucose challenge test to look at glucose levels. And what we have here is fasting glucose, and this is the area under the curve after the glucose challenge test. And as you can see, treatment with our polyphenols significantly decreased fasting glucose levels as well as the area under the curve. We then also looked at inflammation. So this is some of our in vitro studies in placenta and adipose tissue. So as expected, TNF-alpha increases expression and secretion of pro-inflammatory cytokines, and pre-treatment with nobiletin or naringenin can significantly suppress this effect. Likewise, In our mouse model, we have placenta, visceral fat, and subcutaneous fat here. And again, you can see this is, I think this data is representative of a couple of cytokines and chemokines, such as interleukin-1 alpha, MCP, showing that pretreatment with our polyphenols can suppress the inflammatory profile in the mice as well. So, in conclusion, we know that there's increased release of adipokines and adipose tissue-derived exosomes from women with gestational diabetes compared to normal glucose-tolerant women, and these can positively correlate with fetal birth weight. Adipokines and adipose tissue-derived exosomes may mediate the metabolic communication between maternal adipose tissue and the placenta. I've shown evidence that they can induce maternal and placental inflammation. They can cause skeletal muscle and adipose tissue insulin resistance, and they can also alter nutrient uptake. I've also shown some evidence that anti-inflammatory agents such as polyphenols may be promising and novel therapeutic for preventing gestational diabetes. I would just like to acknowledge some of the researchers, particularly the clinical research midwives who collect the samples for us, and the patients who donate all their samples, and all the various funding bodies. Thank you.
Thank you, Marta. I think it's quite a fascinating story, especially the exosome part. So we have time for questions. Probably can start with probably the easy question now, your story about polyphenols. So the amount of polyphenol that you add into your in vitro and your in vivo system, they are similar to, for example, that the concentration that we can find in food, for example. Meaning you can eat whatever, a couple of vegetables or something like that. Yeah.
So it's quite hard to quantify the level of polyphenols because each apple is different in terms of the polyphenols that they'll have. So there's certainly some evidence to suggest it's about, I think, 500 milligrams is about the average that you might get in a diet. And when we compare the doses that we're using in mice, which is 50 mg per kilo, we're probably using a little bit. We're probably using 300 to 500 mg. So it's quite comparable in terms of the concentrations. However, it's hard to quantify really what a person eats.
According to the same question, what kind of polyphenols are you using, or where's the origin of the polyphenols that you're adding to?
So these are just chemical compounds that we buy, like from Sigma.
So you don't
extract
you don't
them.
insulate?
No, I don't extract the polyphenols.
Okay.
So the whole aim is to be able to, in terms of a therapeutic, you want something that's very cheap, that's easily available, that you can just give in a pill form. That would be the hope. So we wouldn't be extracting it from an orange. You would need a lot of oranges. So
As vitamin C. No. It's another question. You don't have proof any property of different fruits or vegetables that have different concentration of polyphenols and different kind of polyphenols in your model.
So we've looked at 2 from citrus fruits here, but we're looking at various polyphenols. So they're just 2. So we've got another 7 or 8 that we're looking at different types from different foods.
Yeah, nice talk. So I was just wondering, so you basically showed that bacteria, probably the LPS somehow, affects the system. In China, there's a very high prevalence of Helicobacter pylori. So I was wondering do you know of any kind of connection there between gestational diabetes and maybe Helicobacter pylori?
No, I don't know if there's any relationship. That's quite interesting. I don't know if there's any relationship, but it certainly fits in with.
Right. Exactly. And the other question I had, I don't know whether you thought about this, but bacteria also produce outer membrane vesicles. So I'm just wondering whether it's actually the LPS or the outer membrane vesicles that might be.
Good point. I don't know. That's a good point. Yeah.
Might be worth looking
Yeah,
into.
maybe worth looking at. Yeah, absolutely.
Thank you, Marta. Just a quick and very simple question, I think. Your exosomes from the adipose tissue were from the obese woman or overweight woman,
So
or a
we
mix?
had a combination. So we had lean, overweight and obese, but they were all matched for BMI. So our two groups didn't differ in terms of their BMI.
Right,
So we had a mix because there were 70 patients in each group, so we had a mix.
and do you think it will be a difference between overweight and obese
We found no
release
relationship.
exosomes?
Yeah, when we looked at the relationship, there was no effect of BMI on content of adipose tissue, of exosomes, sorry.
You mean the content, but you mean the cargo of the exosomes, the number?
numbers,
Ah, right.
yeah.
But then
Yeah.
the cargo may be different, not just the number, I think.
Did we look at the car?
I don't remember, but I think that's a good point actually
Yeah.
in terms of the BMI. And the issue that we didn't find any correlation between the number of exosomes and maternal BMI
Yeah.
was quite a surprise actually. So, and the number we didn't find difference. And for the proteomic analysis,
We didn't separate according to
we didn't
BMI.
because we didn't find any
Yeah.
difference in the concentration. Yeah, but it was very, very interesting actually because previously we already published a paper in which we identify difference in the maternal BMI and the number of circulating exosomes in plasma. So when we performed the experiment, we hoping to get a similar difference, but we didn't find
Yeah,
it.
and we had enough numbers in each group,
Yeah.
so it wasn't a lack of numbers.
And the other important issue when we work with patients, because this is biological sample coming from independent patients, we have a lot of variability also in the groups. So it's something that we need to keep in mind when we perform the analysis.
A very nice talk. Thank you very much. So I have like a question related with the immune system. So during pregnancy, you know, it's a very anti-inflammatory environment for the immune system. So we're supporting like an anti-inflammatory environment. But then you show that in this case, you have a pro-inflammatory cytokines, probably from tissue as well. So I was wondering if you think that the immune system is like sensing these cytokines and said, oh, it's a lot of inflammation, I need to like downregulate this, or the immune system is like playing like a bad role and then said, oh, we have interleukin-1, we're going to differentiate to a pro-inflammatory Th17, for example. Do you have any idea about that?
Well, I think as pregnancy progresses, inflammation increases. So that's been, like in terms of looking at inflammation, it does increase as gestation increases, and that's obviously to allow, cytokines can be good and bad, but I think in that situation it's obviously a good thing, and we think it does that because it directs nutrients away from mum towards the placenta in terms of the studies that we're doing. So I think that what you're probably talking about is early in pregnancy where it's quite an anti-inflammatory state is different to as pregnancy progresses, it becomes pro-inflammatory. And it becomes pro-inflammatory not only for that, but it's an inflammatory state. In order to prepare for labour and delivery, you've got to have inflammation. Inflammation seems to drive labour and delivery, so I think as it progresses, it definitely becomes pro-inflammatory.
Okay, any other question?
Very nice talk. I was wondering about the contents of the exosomes when you did the proteomic. This is from the total exosomes or is a subpopulation?
It's from the total exosomes.
Total exosomes.
Yes.
Okay, probably there are more than one kind or type of exosomes.
Carlos is the expert. As far as I know, there's
sorry.
only one type of exosome.
Yeah.
So this particular exosome coming from adipose tissue explant. So essentially, probably in the explant you have a mix. Probably you have macrophages, you have adipocytes, and you have different types of cells in the explant. So when we get the exosome from the cell condition medium for this plant, we just call total exosome from this plant. But you're totally right. That can be, we don't know if it's coming from the macrophages or other types of cells.
Is that what you mean? Different,
Yeah.
different
I guess, yeah.
cells in the tissue contributing to
those
the different-
exosomes.
Yes,
Okay.
That's
you
our
would have
question.
to obviously separate
Yeah.
the different compartments of the cell, of the tissue. But the issue with that is we just don't get enough tissue to be able
Yeah.
to separate
Yeah, I
the
know.
different populations.
Yeah, I know it's an issue.
Yeah.
That's the reason I asked. is
Yeah.
If you have any way to separate them, I don't know, cell sorting or beads, magnetic bead with a specific marker for subpopulation of exosomes.
Yeah.
I don't
So,
know.
yeah,
Yeah, that's
look,
possible. Yeah.
yeah, I think that is a very good comment. But
Yeah.
also, at least for the aim of the research, we already identify an effect of exosome coming from the adipose tissue, and then we provide the content. So now we need to identify what are the mechanisms associated with the effect, and then probably focus in the microRNA or the protein that are in the exosome.
Yeah.
And that is very interesting approach because everyone working in extracellular vesicles seems to, they identify effect first, and then they identify what in the exosome drive the effect on the target cell, and then they focus in that molecule, and the exosome of the vesicle become a little bit irrelevant. And probably that is the best strategy to continue with the research. Otherwise, you will have the problem about if you have the same content in every single exosome isolation.
Thank you.
Okay. Yeah. Thank you, Marta. Yeah. Okay, fantastic. So we just move forward to our next speakers. So our next speaker, he doesn't need much introduction for us because he was an academic here in our institution before moved to Santiago. Now he's Dr. Luis Sobrevilla, professor of the Pontificia Universidad Católica de Chile. And he's well known not only in Chile, but also worldwide for his research line in endothelial dysfunction, in pregnancy, normal pregnancy, but also gestational diabetes. So yes, I would like to point out something that is very remarkable about Professor Sobrevilla's CVs. He has supervised around 21 PhD students during his career. And it's very remarkable that from these 21 PhD students, the majority of them are still in research with academic position, including myself, because he was my mentor during my PhD. So he has published almost 180 papers in different top journals in the field, and also a wide range of different success funding in the last probably 20 years. So thank you for coming. We're looking forward for your presentation. No, solo es para el streaming.
Okay, thank you very much, Carlos, for the invitation. I'm very happy to be here in Concepcion again. I was in Concepcion, I don't know, 4 or 5 months ago. And it's always very nice to come back and see the scenario. It's very interesting. And also, hello to the professors that are here also, Professor Andrew Quest and Professor Carvajal. Of course, Marta. I'm going to talk about endothelium and placenta, and particularly the alterations that we can see in this type of cells or type of epithelium in the disease of gestational diabetes. And I exclude from the presentation all the data on insulin, pH, adenosine, glucose, etc., because I was told by Carlos that has to be exosomes. So I will show what I can and what I have in exosomes. There was a nice introduction already on gestational diabetes, and this is the general definition of the disease. It's a very bad disease. In fact, in our population, we have in Chile a prevalence of about 5 to 10%, depending on the sector that you are analyzing. For example, at the Catholic University, in Santiago, you can reach up to 20% of prevalence of gestational diabetes. But in Chile, up to 20%. But these values in Chile are very close to what we can see worldwide. And the disease is bad, of course. We all know about that because the original definition is a disease that is of the mother, in fact. However, we now know, and after, I would say, 20 years of research by different groups around the world, that there are many perinatal risks, like for example, microsomes, as it was already mentioned. In some countries, still we have fetal death, which is interesting to see. Chile is not seeing this kind of complications, likely. But also we have postnatal risks like obesity, gestational diabetes in the woman that was born from gestational diabetes, hypertension, dyslipidemia, and type 2 diabetes mellitus. So it's a disease of pregnancy that is also associated with the concept of a disease of the mother that we now know is a disease of the mother and the fetus, and of course the placenta. So I want to show you some data that the placenta is also altered in this disease. The placenta is holding the fetus, of course, and there is a very complicated network of vessels, and microvessels and macrovessels are composing this tissue, or this organ, in fact. And the most used cells of the representative of the macrovasculature are the human umbilical vein endothelial cells in this umbilical cord, and also the umbilical artery endothelial cells. And then from the microcirculation, we have this villi in here where the blood of the mother is very close. Something happened. It's very close to the circulation of the fetus, but they're not getting in touch because the barrier separating is syncytial trophoblast, and the proper endothelial cells from the microcirculation. So we have the human placenta microvascular endothelial cells as representative of this very interesting tissue of the microcirculation of the placenta. Lopalish in 2010-2011 published a series of papers, one of these in placenta, highlighting the differences between micro and macro circulation and the properties of the endothelium of these two different vascular beds. And she made a very nice contribution to the field to state that the macro circulation and microcirculation will have different properties to respond to give an answer to different stimuli, like, for example, insulin and other. And the cells, we can isolate the cell from the endothelium from this vessel, from the macro and the micro vessels, and this is a way that the cell could look at. This is a very simple picture in a light microscope that was made by Felipe Sangüesa, a former student from the university. It's not very technically accepted, but it's very nice, I think, because you can see individual cells, you know, with a body that we normally imagine. But this picture was taken 15 years ago, perhaps, and it's very nice. So I like it. I'll show you. From these cells, we can expect the release of several molecules that will have a direct action on the underlying smooth muscle cells. So to end in contraction or dilation of or relaxation of the smooth muscle cells. And then in that way, the endothelium is regulating the vascular tone and then the blood flow from the placenta circulation to the fetus and vice versa. And these molecules, for example, are the nitric oxide, of course, the most known gas with vasodilatory properties, several amino acids, and one of these, of course, the source for the production of nitric oxide, the cationic amino acid L-arginine, and other molecules a little bit more unknown in the literature, but now a little bit more considered for several groups. This nucleoside, adenosine, and not only the nucleotides, ATP, ADP, and AMP even, but now also adenosine as a key regulator of the vascular function in the human placenta. So the regulation of the capacity of these cells to synthesize and release or degrade the different molecules that are acting on the smooth muscle cells and then regulating the blood flow are very, very important. Very early in the literature, we published, in fact, in 1995 already. Sorry, I got a little depressed with that. We published that the uptake of adenosine by the human umbilical vein endothelial cells was altered in gestational diabetes, and it was reduced, in fact, via these membrane proteins N1 and N2, and also later on, not too much later, 1 year later, we published that another molecule, L-arginine, which at that time were completely different in the field of research. One group was studying adenosine and nucleosides, and different groups were studying cationic amino acid and other amino acids. But then we saw that also L-arginine transport, in this case mediated by the cationic amino acid transported type or subtype 1, was altered also in gestational diabetes, and in this case was increased. So that was interesting to see that two substrates, but was not very much interesting for other people because there are two different substrates, and the uptake could be up or down or not modified. But what we saw as an interesting finding here is that in fact adenosine and L-arginine are two key molecules to generate vasodilatation in the human placenta vasculature. And the mechanism by which adenosine and L-arginine does it is by increasing nitric oxide synthesis and release and bioavailability, in fact. So some relationship was between L-arginine and adenosine at that time that we all know a little bit more now. Another observation is that in gestational diabetes mellitus, the umbilical vein plasma adenosine concentration was about 3 times higher in gestational diabetes compared with blood from normal pregnancies. So this umbilical vessel is bringing blood to the fetus with a large concentration of adenosine very far, I would say 3 times from normal circulating adenosine. This is perhaps coming from the placenta, since the concentration, the elevated concentration of adenosine was only seen in the umbilical vein, but not in the umbilical arteries, suggesting that the alterations to increase the concentration of adenosine in the umbilical vein was mainly due to an alteration in the placenta metabolism or handling of the adenosine or nucleotides releasing adenosine. In fact, adenosine increased in the umbilical vein is important because it has also the ability of vasodilate the umbilical vein, like is shown here. Adenosine is generating vasodilatation in the human umbilical vein, vein rings in a myograph. And it was published by Francisco Estermeyer in 2011 as the first time in the literature for fetal placenta circulation. What is the source of this adenosine? There are several sources, and one of these is that adenosine generated intracellularly can be released to the extracellular space by these nucleoside transporters working in an equilibrated manner leading to a similar concentration between extracellular and intracellular of adenosine. But the other source are of course, the degradation of AMP by CD73 into adenosine at the extracellular side of the plasma membrane. So we have ectonucleotidases that are releasing adenosine. And interestingly, even when we have these adenosine deaminase at the plasma membrane in endothelial cells, the activity and the affinity of adenosine deaminase for adenosine is very, very low. And even when adenosine can be converted into inosine, the efficiency of this process is very low. So whenever you have a degradation, increased degradation of AMP to adenosine, the accumulation of this nucleoside will be the result of the incapacity of the adenosine deaminase to degrade and keep a physiological concentration of adenosine extracellularly. So the increased adenosine concentration that we were seeing in the umbilical vein blood could be also because of overactivity or expression of CD73 in these cells. However, other cells from the blood, for example, the platelets, or other mechanisms like shear stress in the vasculature could also lead to the release of adenosine from the endothelium. Since adenosine is accumulated extracellularly, the connection between adenosine and L-arginine was then published in 2006 by Rodi San Martin, who is now in the south in Austral University, and later on by Pardo, Fabian Pardo. Sorry, it's the age, I know him. I work with him every day, you know. In 2013, recently, he put all together the data on accumulation of adenosine extracellularly in gestational diabetes, and by a different mechanism, by specifically acting on A2A adenosine receptors, lead to an increase in an arginine entry and in its metabolism to nitric oxide by the synthase of nitric oxide, or nitric oxide synthase from the endothelium. And this accumulation was explained by the reduced expression of the genes coding for the N1 transporters mainly in the macrocirculation due to activation of transcription factors like, for example, CHOP complex of protein that will react also in a condition of endoplasmic reticular stress, for example, in these cells. So in gestational diabetes, we have accumulation of adenosine due to reduction in the expression and activity of N1, leading to stimulation of a signaling pathway that also include the overexpression of the gene coding for HCAT1, and then increasing the uptake of this amino acid to end with the overproduction of nitric oxide in these cells. This overproduction of nitric oxide will then keep going, stimulating different pathways, like, for example, P44, 42 MAP kinase, and activating again this inhibitory pathway that finally will result in increased adenosine concentration extracellularly. So here is the L-arginine uptake increase and the adenosine uptake reduced in gestational diabetes in a more comprehensive and functional manner. In fact, these cells from gestational diabetes are very much good in releasing nitric oxide, for example, as measured here by fluorescence by Marcelo Gonzalez, Professor Gonzalez from the Faculty of Biological Sciences here. But more interesting than that, I will say, was that the transcription factor CHOP in complex with the CEBP alpha molecule are in fact binding to a specific sequence in the promoter of the gene coding for HN1 in these cells. And the binding in gestational diabetes is increased, resulting in a reduction of the expression of this gene. So the cells from gestational diabetes are synthesizing more nitric oxide. The nitric oxide will activate these transcription factors, and the factors go to the promoter to bind here and reduce the expression of the gene. If we analyze the binding now in cells that are not expressing or are very much down expressing the INOS, the nitric oxide synthase, endothelial nitric oxide synthase, the binding in gestational diabetes, which was upregulated, is back to normal or even a little bit lower. So binding of CHOP, CEP alpha complex to the sequence of the SLC gene is dependent on nitric oxide in these cells. And these cells, remember, are releasing a lot of nitric oxide by the mechanism that I just showed you. Marcelo Farias in Santiago summarized this data showing exactly this. The combination of these transcription factors are stimulated then or increased in activity by nitric oxide to bind to a specific sequence in the promoter in that region, reducing the expression and then accumulating adenosine extracellularly. But what are the factors involved? And here, I love to talk about pH and insulin and everything, but I was told not to do it. So I'm going to show you something about this, exosomes, which is a very, in fact, not preliminary, but published already, but still very starting study on this. As was mentioned already, the placenta and other tissues are releasing, of course, exosomes with the different contents or cargos, and it was mentioned that microRNAs are the main cargo and other proteins, which are still being identified from different tissues. We can see long lists of proteins and other molecules in the cargo of the vesicles, and this, I expect, it will grow even more. And the exosomes are only one type of extracellular vesicles that we have, of course, for example, in placenta, apoptotic bodies, syncytial nuclear aggregates, and plasma membrane, the proper extracellular vesicles, more than in the size of nanometers. And then we went to the placenta, and we asked a different question that was in the literature. The literature question was, is the placenta releasing exosomes? And are these exosomes altered in number or activity, whether they're coming from a disease of pregnancy, like gestational diabetes, as Carlos has published, and others, as Marta, or in other pathologies like preeclampsia, or pathologies affecting, for example, the fetus directly in a very evident manner, like intrauterine growth restriction. And then we decided to look at the ability of the human umbilical vein endothelial cells, freshly isolated, of course, from gestational diabetes, to generate and release exosomes. So we did the proper analysis. I'm not going to go through that. But then when we separate different fractions from the culture media that we isolated from there, we have a fraction which is positive for the markers for exosome. We did, in collaboration with Dr. Carvajal in his lab in Santiago, the characterization of the size and the shape, and we thank him all the time for that. Then we look at the fraction 11 of this centrifuge differentiation. How do you say that? Centrifugation. Okay, and the fraction 11, it was positive for these markers in both type of nanovesicles, from the normal and from the GDM pregnancies. And the question was, is it able, or are these vesicles able to modulate the phenotype of normal cells if they're coming from gestational diabetes, or whether they are able to modify the phenotype of a gestational diabetic cell that we call is a ill cell, whether they are coming from a normal pregnancy. It's like having a good friend or a bad friend when I'm a good friend or a bad friend. Do you understand? So we isolate exosome from normal pregnancies or gestational diabetes, and we did the cross experiments in cells from normal or GDM pregnancy. One of the main tools used in the literature is, of course, to look at the ability of these cells to regenerate in a wound healing assay, the space. And I'm just going to show you here, these are cells from normal pregnancies exposed to exosome from normal pregnancies, no? Or from exosomes exposed to from gestational, isolated from gestational diabetes pregnancy in different concentrations for normal cells or for GDM using this scheme. We can see numbers, of course, and the numbers are just to mention to you, this is it's an expression to measure the time required to repair the wound healing in an assay. And in normal cells, in control conditions, this is a factor that we calculated by an equation published by Tamara Sáenz this year, in fact, at the beginning of this year. And if we look at the effect of exosomes from normal pregnancies in cells from normal pregnancies. The value is very similar, even at 2 different concentrations. But now, if we incubate these cells from normal pregnancies with exosomes from gestational diabetes, the value is really higher, indicating that the repair period was longer than normal. So that's the observation. But the conclusion is that these exosomes are saying something to these cells or doing something to these cells to repair the wound. Wound, you say? Wound. Slowly, okay? And if we go now to GDM pregnancy and isolate the cells and do the assay, we have a longer time to recover the same area. If we incubate with gestational diabetes, derived exosomes is almost the same, right? But now if we incubate the cells with exosomes from normal pregnancies, the 0.1 value is reduced about 30% or 20%, significantly. So in this case, we can see this, in fact, that the GDM isolated from gestational diabetes will change perhaps the phenotype for this assay in cells from normal pregnancies, making them very similar to what we see in the cells from gestational diabetes. And on the other hand, we can also see that the incubation with exosomes from normal pregnancies of cells from gestational diabetes, this is not kept or increased, but in fact is back to a value close to normal pregnancies. So the gestational diabetes condition is making that the endothelial cells, particularly HUVECs, are releasing exosomes that will change the phenotype of a normal cells, at least in vitro, and vice versa. If we look at now for transport of L-arginine, which is one of the substrates to synthesize nitric oxide and then modulate the proliferation of these cells in vitro, let me just ask you to concentrate in this green symbol here for control, which is this. This is in the uptake in different concentrations of L-arginine in the extracellular space, and with a Vmax of approximately 0.8 or 1 picomole per microgram protein per minute for the uptake of L-arginine in cells from normal pregnancies. If these cells are now incubated with exosomes from gestational diabetes at 3 different concentrations, I will look at the 0.5 first, the blue symbol here. We can see there is an increase in the Vmax of about 1.2 Vmax for picomoles for the Vmax of arginine transport. Now, if we incubate the cells with a similar concentration of exosome from normal pregnancies, which is that light blue, is almost the same as the control value. If we go now to gestational diabetes, the green symbols here, again, are very much higher compared with the values for normal cells. You know, it's about double the Vmax, about 1.6, 1.8. And this phenotype is back to normal values when we incubate the cells with exosome from normal pregnancies. For example, the light blue here. You can see this is without the exosomes, and this is with exosomes from normal pregnancies. This is the table for that. I don't want to see that. But just to show you that the ratio of Vmax over Km as an index of the maximal transport capacity of the phenomena that we are seeing with the substrate, in this case arginine, gestational diabetes changed this phenotype into a very similar one to the pathology, and the phenotype in the pathology is back to not to normal, but is reduced significantly from a gestational diabetes phenotype to more a normal phenotype. So we are seeing also this phenomena in a very specific mechanism, the uptake of L-arginine. If we look at and associate, I just wanted to mention that the overall transport here is unaltered by any conditions, even in normal and in gestational diabetes pregnancy, suggesting that perhaps one or two mechanisms for the 5 different mechanisms of transport for elageny in these cells is only affected by the exosomes, but no other mechanisms. So this is a more kinetic study, but I don't want to disturb you with that. If we look at that, we then decided that, or concluded that it is most likely that the HCAT1 transporter was altered, since the kinetic values and because of the overall satural transport was unaltered by the exosomes. And then we look at the expression, of course, the HCAT1 in the cells that were challenged with the exosomes. And what we see in cells from normal pregnancies is that this is the relative value to almost 1 in a series of experiments. Incubation with normal exosomes does not alter the protein abundance, but incubation with exosomes from gestational diabetes really increases. And if we look at the cells from gestational diabetes, there is already a huge increase compared with normal. And then this phenomenon is back to almost normal when you incubate the cells with exosomes from normal pregnancies. And even when you incubate with gestational diabetes, you have a reduction, but it's still significant from controls. So the arginine transport capacity, maximal transport capacity in this is perhaps associated with the alterations in the expression of protein abundance for this transporter. If we look at now at the synthesis of nitric oxide, the endothelial isoform, under the same conditions, we can, here is just the blot, but we look at the activator phosphorylation, serine 1177, or the inactivator phosphorylation of the enzyme threonine 495, and of course the total one, and under the same condition. And we saw exactly the same, no effect for exosome from normal pregnancies in cells from normal pregnancies, but an increase when the cells are exposed to gestational diabetes. This is the increase in the total protein for gestational diabetes, which is reduced by exosome from normal pregnancies again, and unaltered by exosome from gestational diabetes. If we move at to see the serine phosphorylation or the threonine phosphorylation, we found something very interesting, that is the increase caused by gestational diabetes in the total protein. It was not seen for serine activation, so we are seeing a change in the amount of protein instead of the activator phosphorylation of the enzyme. And we were then, only with this data, expecting not to see any change in the synthesis of nitric oxide, or perhaps a similar increase to the increase in the amount of protein in these cells. But I will show you later. If we look at now at the gestational diabetes serine phosphorylation, it is increased compared with normal cells. Exosome from normal pregnancies reduce it back to a normal value, and from gestational diabetes are not really altered. Looking at the inhibitor phosphorylation now of phenos, there is no change in exosome from normals, but look at here, exosomes from gestational diabetes are reducing the phosphorylation in the inactivator phosphorylation in threonine in enos. In that case, when you have less inhibition, you have more activity. Right? It's like not having the parents at home when we have a party. If we look at now at gestational diabetes, nothing happened with the threonine. So perhaps gestational diabetes cells and the activity of exosomes to threonine 495 inactivator phosphorylation is not really involved in this phenomenon. Looking at the synthesis of nitric oxide, in fact, we can see that the exosomes from gestational diabetes are increasing the level of nitric oxide, and this increase in gestational diabetes is reduced by exosome from normal pregnancy, but not from gestational diabetes. But this increase in the NO detected here might be due to a reduction in the inhibitor phosphorylation of phenos instead of a change in the activator phosphorylation of phenos inside. Interestingly, if we look at the ROS generation. We have the data for specific superoxide anion, but this is just a ROS here. We can see that gestational diabetes exosomes are increasing the ROS concentration in the medium. And this is very interesting because you have cells from gestational diabetes that we are seeing we are seeing a more release of nitric oxide. But if we consider this data now in cells from normal pregnancies incubated with this, where you have more protein and more synthesis of nitric oxide, perhaps it is expected to have even more than what we are measuring, because these ROS are going to scavenge the nitric oxide generated in the medium. So we have to correct that. We have to analyze how this increase in the ROS level will, in fact, be reducing what we were going to see in the nitric oxide level in these cells. So this is just to show that this effect is seen in normal cells, but not in gestational diabetic cells. Interesting. So as a summary, QX release exosomes. Yeah, the exosome from gestational diabetes, we believe, is causing a phenotype in normal cells that we are calling a GDM phenotype, based on what we are looking at: arginine transport, nitric oxide, and the capacity to repair the wood. And the exosomes from gestational diabetes also, of course, increase this signaling pathway, arginine nitric oxide, in these cells. I'm not finished. Do I have another?
Sure.
Or do you want me to finish? If we move now to the placenta, and I want to show you some data that is not published. It's done by Indira Chiarello, a postdoctoral in the lab. She asked in her postdoctoral project whether the exosomes that are being generated by the syncytial trophoblasts in the placenta are being released, which we know, by the apical membrane to the maternal circulation, whether those exosomes are the same, or these exosomes are coming back to the fetal circulation, or whether these exosomes released by the basolateral membrane in these syncytial trophoblasts are different from the other one. So rapidly she did the isolation, but then she cultured the cells in this matrigel, 2 chambers. So you have the separation of the apical membrane from the basal membrane, and the medium here is different, not in contact with the basal medium, as you know. Then to test whether you have a monolayer or syncytium, in fact, of high resistance, we look at the using the T. How do you spell T? The trans-epithelial electrical resistance. Okay, and then we put an electrode out in the apical medium and the basolateral medium, and we look at the resistance there with the help of Dr. Carvajal. Looking at the day 3 or day 5 of the isolation of the syncytial trophoblast. Sorry for that, but this is cytokeratin 7 just for the trophoblast. Okay, thank you. Thank you. This is the merge for DAPI and the cytokeratin 7. You can perhaps see that is you cannot see very much, but it's very clean that there is a monolayer. And in fact, if you look at the sagittal pictures here, you have a continuity in the signal on this side and on this side. But in day 5, if you do the same, you have very large gaps between the layer, indicating that you have very much holes in here. Like, for example, this one, if you can see that, but if not, just believe me. But this is, in fact, after day 5, the expected resistance or T value will be very low because there is no resistance in there. So we measured that, and what we saw is that the first day nothing happened, but then at days 3 to 4, there is a very nice and maximal T value, and then at day 5 it's down back to 0. So we isolated the exosomes then in this region, day 3 to 3.5 in culture. So we were sure that we have a monolayer, and the exosome from the apical will not go through these holes in the cells to the other side. Well, okay, this is the shape from the apical and basal. There is no difference in the amount and the size of the vesicles. These vesicles can bind at least to microvascular endothelial cells. And if you look at the vesicles now from apical or basal membrane, forget this is the same, but you can look at here these green dots for the vesicles labeled with this green protein called PKH67. You can see some dots in the endothelial cells. Yeah, there's the nucleus. And if you look at the basolateral membrane, you have much more supposedly binding to the cells in these microvesicles. So the vesicles from the apical side are able to bind to endothelial cell, but the vesicles from the basolateral side are more able to bind to the endothelium. So is this really a difference only in the dynamics of binding, or it will have a consequence to the cells? Since the release of adenosine is a mechanism altering gestational diabetes increase, and then the next question was whether the increase that we are seeing in the placenta circulation, looking at the umbilical vein blood, for example, as I showed you before, the question was, is the exosome really released by the basolateral side of the trophoblast contributing to increase the concentration of adenosine in this vascular bed? So we look at the CD73 in these vesicles, and what we found in the apical membrane, there is no CD73, but at the basolateral vesicles, we have a nice signal for the CD73. This is just another experiment. So these extracellular vesicles, perhaps nanovesicles, are containing the CD73 only in the basolateral membrane, which could contribute to generation of adenosine. And now if we incubate the exosomes with AMP as a source of adenosine, we can see the peak of adenosine when the AMP and exosomes are there, and a reduction in the AMP level in this in an HPLC pattern. This is the AMP, the blue one, if you can see, and the basal exosomes without the substrate is in black. So these vesicles from the basolateral side of the syncytial trophoblast express the CD73 and can degrade AMP to adenosine. Now we have to see what happened in gestational diabetes. Unfortunately, we don't have the data yet, but it will. So as syncytial trophoblast release exosomes, this might be different from both sides. This from the basolateral side may generate adenosine and contribute to the increased adenosine seen in gestational diabetes. We have to check that. So I'm just finishing. I just want to point out that in our country, and I guess in many other countries also, the obesity according to the report by the MinSal in November last year, half of the children are overweight in our country, and 25% of the children are obese, and the prevalence of gestational diabetes or diabetes is increasing along the time. And please consider that diabetes and obesity are problems during the maternal, during the pregnancy, that will affect the children and the young and adult life, and of course elderly life. But then this will have a consequence again from women that are going to get pregnant and that were born from these conditions. In our country, in our hospital in Santiago, we have this distribution of samples. These are only 204 samples. Look at here. The BMI is over 30 in a large proportion. And this was expected many years ago, not many, but 4 years ago by Professor Araya at the University of Chile. A large increase. Now we can put in here the other value. And if you look at the girls that are in the reproductive age, you will see that from 2003 to 2010, the prevalence of overweight is increasing this proportion, and from 2010 and 2017, only 7 years, again, is still increasing, another 9%. And if you look at the obesity in women in their reproductive age in Chile, it's increasing 23% in the last 7 years. So what we are going to see in the next coming years is an increase in women that are pregnant that were obese before pregnancy. And that is a bad condition. This is what was asked by Roberto Villalobos, also from here. He studied biochemistry here. And he said, I'm going to look at the alteration in these signaling mechanisms that we know in women with the BMI at the pre-pregnancy BMI in the range of obese compared with normals. And what he is claiming now in a nice paper middle of this year is that there is endothelial dysfunction due to endoplasmic reticulum stress when the mother is with pre-pregnancy obesity. So that we are going to see. This is the mechanism. There is a lot of other information, and I wanted to just show it because CHOP is increased as one of the key molecules addressing ER stress, and also JANK1 as a mechanism for regulation of insulin signaling in the cells. Tamara Science, in fact, published in 2018 at the beginning that the phytoplacental exosomes will regulate L-arginine transport, nitric oxide synthesis, and adenosine transport. So the pathway that we were discussing before in gestational diabetes. We know about type 1 diabetes mellitus, type 2 diabetes mellitus, and the exosomes and microvesicles, and the content of this leading to endothelial dysfunction. But we don't know very much about gestational diabetes, the effect of exosomes in the fetal circulation to lead to fetal endothelial dysfunction. So perhaps that diabetes that we are seeing in patients that are obese and develop type 1 diabetes mellitus, adult subjects, can also be seen in gestation, during gestation with a condition of gestational diabetes mellitus in women that were with pre-gestational maternal obesity, and what we are calling gestational diabetes, which is just now accepted by Roberto in Molecular Aspect of Medicine, publishing in February next year. Are these exosomes also carrying something else? I mean, not just reaching the cells and producing something in the cells, or are these exosomes being a kind of communicating an epigenetic modification that will have a consequence in the mother, in the placenta, and in the newborn as these molecules are changing. So we cannot keep going, I will say, thinking in exosomes as the molecule that will alter a mechanism now in that moment. What will be the consequence of it on other phenomena? And epigenetic is one. And insulin resistance, as it was mentioned today, insulin resistance via these mechanisms involving nitric oxide again, and for example, NSCAP-B and ROS, could also be altered by extracellular vesicles as proposed by Tamara Science in a paper just accepted also. Just finished, okay? Exosomes and GDM is a matter, yes, exists. The fetoplacental vasculature is releasing the exosomes very active and very badly, I would say sometimes. This concept, the basolateral, basoapical, I think is important to keep in mind because I know Professor Sadowski in the States is also doing something similar in the rat model. And, but we have to think in the long term of the effects of exosomes or extracellular vesicles, not just the moment that we are seeing it. Just finishing, this is the group. In fact, it's a very old picture. I have to renew it, but this is Indira Sciarello, who was mainly acting on a placenta. And this is me with tie. And I want to say thank you to Tamara. Tamara Sach is now in Canada doing the postdoctoral. She was the first person in the lab that was working in exosomes, and I didn't want to do it, but she wanted to do it, and fine, she did, very nice. And Carlos, of course, he was the first guy going to Australia with Greg Rice to learn from the lab, and I'm very proud of you. And the postdoctor in the area. So, and of course the money from the CONICIT and other sources. Thank you very much.
Thank you. Okay, so we have time for a couple of questions or comments. After that, we have the coffee break for 15 minutes, and then we have the presentation from Andrew before the lunch break. Probably I can start with just a quick comment about your migration assays. Probably is an easy word to. Can you mention something about the timing? Because it's very interesting when you incubate the cell with the exosome, you have an effect in terms of the bioactivity and the transport. So what do you think in terms of the delivery of bioactive molecule to these cells? And this is an acute response that can happen in the first, 2 to 3 hours, or there is some molecules inside of this exosome that will affect the translation, for example, protein or transporting into the membrane.
Yeah, I didn't mention that, but there are two set of results. One is from 0 to 4 hours, and the other one is from 0 to 8, or if you want to say 4 to 8 hours. And the effect in 0 to 4 hours is maximum, I would say. 4 to 8 hours is already not very significant from 0 to 4 hours. So the effect of exosomes that we are seeing in that assay is in the first 4 hours. Now, whether this will mean something in the changes in the expression of protein or activity of promoters, yes, it will, because for example, the arginine CAT1 could be increased in the expression in about 3 hours for other molecules. I don't know Vesicles exactly, but other kind of responses. For example, insulin, you can see an effect on 8 hours, but the maximal effect at 8 hours. But then you have a half maximal effect of insulin in about 2 to 3 hours for HCAT1. So it could be that the effect is on that specific time period, and because we are not seeing any other change in the assay after 4 hours to 8 hours, it could not be. But we don't know what really is happening between 4 to 8 hours.
Mark?
So your exosomes, have you characterized the content in your exosomes, and do you know, can you hypothesize what's causing the changes that you're seeing?
No. No, we haven't characterized it, and it's an open question, and I don't know, I'm not a biochemist, I'm not a biophysics, yeah. And then what I really want to see now is really whether these exosomes that we are isolating, will they have an effect in some assay? They have. So now we have to try to identify what is in there. But my question is, do I want to identify 200 molecules? Or do I want to ask the exosomes whether the increase in for example, the HCAT1 protein abundance in cubics is because the exosomes were carrying already the HCAT1s and then just adding to the plasma membrane, as we saw in initial experiments for insulin receptors, for example, insulin receptor A and B, which I didn't show here. If you look at the exosomes isolated from cells knocked down for insulin receptor A or B, you don't see in the target cells the insulin actions looking at the signalings. So it is very likely that the exosomes are carrying the proteins that we are seeing overexpressed or in high quantity. That's why I say protein abundance more than expression.
Yeah, maybe I missed it, but so you mentioned that these exosomes from gestational diabetes, right? They reduce the migration in your migration assay.
They delay it.
Yeah, delay. And so they then increase basically arginine uptake, right? So is there a correlation?
Yeah.
Is there a connection between arginine uptake and migration?
There is a connection, but on the other way, because if you look at we did this in cubic. If we look at cubic and the effect of a donor of nitric oxide, the effect, in fact, is increasing. No, sorry, it's reducing the proliferation of cubics in vitro. In fact, if you look at the time needed to reach the confluence, maximal confluence in vitro for cubics from gestational diabetes is double the time than normal cells. And if you do a knockdown for those cells for enos, then the displacement in the graph to the right side is back to normal cells. So we think in cubics that nitric oxide is reducing the proliferation. You see? So at the end of the day, the exosomes isolated from gestational diabetes will come to normal cells and will offer to the cells more NOS as a protein, as is already published, in fact, from trophoblast, not from cubics. And then the cell will have the more protein abundance to produce nitric oxide and then delay the repair of the problem in the assay.
And the other question I had is, I mean, so it seems like NO production is really important in what you're seeing. So could you just treat this with L-name or something?
Yeah.
Some kind of NOS inhibitor?
Absolutely.
Everything would go away?
Absolutely. Yes, we did that. We have a paper published also in placenta this year for those kind of assays, but using high glucose, extracellular glucose, because we didn't have the EDM samples yet. We have a set of results, but we published first the high glucose and using a name and the enos knockdown. And in fact, it correcting the problem. Yeah.
Okay, so thank you. That was
Question.
of course.
So your GDM cohort, are they treated with insulin or diet? Are they obese or non-obese?
Not the question.
Yeah, the patients that I show here in BBA's report from Tamara, they are not treated with insulin. They are only treated with diet, and they are all obese, not overweight.
All obese.
All obese.
And your controls, are they?
They are lean. They're normal.
So do you
Normal
think
weight.
it's an effect of gestational diabetes or obesity?
No, we don't know. We are now separating the data, and this is a message for the new students, our new researchers, is that the obesity cannot be now, I mean, the samples from whatever you took from an obese woman, you have to separate from obese and overweight. We presented the data 2 weeks ago when a student presented in the Neurophysiology meeting in London, and he showed that the alteration that insulin could cause in cells from obese woman will be different from overweight woman with gestational diabetes. So it's a very key point. And I'm reviewing my own publications, trying to separate those group of data, but it's impossible now. Not now. No, impossible, because there was all mixed. In fact, Dr. Guzmán Gutiérrez published a very nice paper, I don't know how many years now, a few years ago. And his data is mainly obese, and if not all obese. It's one of the first papers where we separated the obese from the overweight. And because we are claiming that the gestational diabetes will be a condition, it cannot be gestational diabetes in mixed obese overweight. It has to be only obese. So we are looking for that.
Okay, fantastic. So thank you again.
Thank you.